Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Golgi apparatus. 76 languages. Diagram of a single stack of Golgi. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. Not to be confused with girification. For the song, see Junta, album. The Golgi apparatus, /LDI/, also known as the Golgi complex, Golgi body, or simply the Golgi, is an organelle found in most eukaryotic cells. 1. Part of the endomembrane system in the cytoplasm, it packages proteins into membrane-bound vesicles inside the cell before the vesicles are sent to their destination. It resides at the intersection of the secretory, lysosomal, and endocytic pathways. It is of particular importance in processing proteins for secretion, containing a set of glycosylation enzymes that attach various sugar monomers to proteins as the proteins move through the apparatus. The Golgi apparatus was identified in 1898 by the Italian biologist and pathologist Camillo Golgi. 2. The organelle was later named after him in the 1910s. 2. Discovery. Owing to its large size and distinctive structure, the Golgi apparatus was one of the first organelles to be discovered and observed in detail. It was discovered in 1898 by Italian physician Camillo Golgi during an investigation of the nervous system. 3. After first observing it under his microscope, he termed the structure as apparato reticolare interno, internal reticular apparatus. Some doubted the discovery at first, arguing that the appearance of the structure was merely an optical illusion created by the observation technique used by Golgi. With the development of Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. 3D rendering of Golgi apparatus. Modern microscopes in the 20th century, the discovery was confirmed. 4. Early references to the Golgi apparatus referred to it by various names, including the Golgi homegrown apparatus, Golgi homegrown ducts, and Golgi Kopsch apparatus. 2. The term Golgi apparatus was used in 1910 and first appeared in the scientific literature in 1913, while Golgi complex was introduced in 1956. 2. Subcellular localization. The subcellular localization of the Golgi apparatus varies among eukaryotes. In mammals, a single Golgi apparatus is usually located near the cell nucleus, close to the centrosome. Tubular connections are responsible for linking the stacks together. Localization and tubular connections of the Golgi apparatus are dependent on microtubules. In experiments it is seen that as microtubules are depolymerized, the Golgi apparatuses lose mutual connections and become individual stacks throughout the cytoplasm. 5. In yeast, multiple Golgi apparatuses are scattered throughout the cytoplasm, as observed in Saccharomyces cerevisiae. In plants, Golgi stacks are not concentrated at the centrosomal region and do not form Golgi ribbons. 6. Organization of the plant Golgi depends on actin cables and not microtubules. 6. The common feature among Golgi is that they are adjacent to endoplasmic reticulum, exit sites. 7. Structure. In most eukaryotes, the Golgi apparatus is made up of a series of compartments and is a collection of fused, flattened, membrane-enclosed discs known as cisternae, singular. Cisterna, also called dictyosomes, originating from vesicular clusters that bud off the endoplasmic reticulum. A mammalian cell typically contains 40 to 100 stacks of cisternae. 8. Between 4 and 8 cisternae are usually present in a stack, however. In some protists as many as 60 cisternae have been observed. 4. This collection of cisternae is broken down into CIS, medial, and trans compartments, making up 2 main networks. The CIS Golgi network, CGN, and the trans Golgi network, TGN. The CGN is the first cisternal structure, and the TGN is the final, from which proteins are packaged into vesicles destined to lysosomes, secretory vesicles, or the cell surface. The TGN is usually positioned adjacent to the stack, but can also be separate from it. The TGN may act as an early endosome in yeast and plants. 6. 9. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. There are structural and organizational differences in the Golgi apparatus among eukaryotes. In some yeasts, Golgi stacking is not observed. Pigia pastris does have stacked Golgi, while Saccharomyces cerevisiae does not. 6. In plants, the individual stacks of the Golgi apparatus seem to operate independently. 6. The Golgi apparatus tends to be larger and more numerous in cells that synthesize and secrete large amounts of substances, for example, the antibody-secreting plasma B cells of the immune system have prominent Golgi complexes. In all eukaryotes, each cisternal stack has a CIS entry face and a trans exit face. These faces are characterized by unique morphology and biochemistry. 10. Within individual stacks are assortments of enzymes responsible for selectively modifying protein cargo. These modifications influence the fate of the protein. The compartmentalization of the Golgi apparatus is advantageous for separating enzymes, thereby maintaining consecutive and selective processing steps. Enzymes catalyzing early modifications are gathered in the CIS face cisternae, and enzymes catalyzing later modifications are found in trans face cisternae of the Golgi stacks. 5. 10. Function. The Golgi apparatus is a major collection and dispatch station of protein products received from the endoplasmic reticulum. Proteins synthesized in the are packaged into vesicles, which then fuse with the Golgi apparatus. These cargo proteins are modified and destined for secretion via exocytosis or for use in the cell. In this respect, the Golgi can be thought of as similar to a post office, It packages and labels items which it then sends to different parts of the cell or to the extracellular space. The Golgi apparatus is also involved in lipid transport and lysosome formation. 11. The structure and function of the Golgi apparatus are intimately linked. Individual stacks have different assortments of enzymes, allowing for progressive processing of cargo proteins as they travel from the cystine to the trans-Golgi face. 5. 10. Enzymatic reactions within the Golgi stacks occur exclusively near its membrane surfaces, where enzymes are anchored. This feature is in contrast to the which has soluble proteins and enzymes in its lumen. Much of the enzymatic processing is post-translational modification of proteins. For example, phosphorylation of oligosaccharides on lysosomal proteins occurs in the early CGN. 5. Cis cystine are associated with the removal of mannose residues. 5. 10. Removal of mannose residues and addition of N-acetylglucosamine occur in medial cystine. 5. Addition of galactose and sialic acid occurs in the trans-cystine. 5. Sulfation of tyosins and carbohydrates occurs within the TGN. 5. Other general post-translational modifications of proteins include the addition of carbohydrates, glycosylation, 12, and phosphates, phosphorylation. Protein modifications may form a signal sequence that determines the final destination of the protein. For example, the Golgi apparatus adds a mannose. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The Golgi apparatus, salmon pink, in context of the secretory pathway. Diagram of secretory process from endoplasmic reticulum, orange, to Golgi apparatus, magenta. 1. Nuclear membrane, 2. 6-phosphate label to proteins destined for lysosomes. Another important function of the Golgi apparatus is in the formation of proteoglycans. Enzymes in the Golgi append proteins to glycosaminoglycans, thus creating proteoglycans. 13. Glycosaminoglycans are long, unbranched polysaccharide molecules present in the extracellular matrix of animals. Vesicular transport. The vesicles that leave the rough endoplasmic reticulum are transported to the CIS face of the Golgi apparatus, where they fuse with the Golgi membrane and empty their contents into the lumen. Once inside the lumen, the molecules are modified, then sorted for transport to their next destinations. Those proteins destined for areas of the cell other than either the endoplasmic reticulum or the Golgi apparatus are moved through the Golgi cystine towards the trans face, to a complex network of membranes and associated vesicles known as the trans-Golgi network, TGN. This area of the Golgi is the point at which proteins are sorted and shipped to their intended destinations by their placement into one of at least 3 different types of vesicles, depending upon the signal sequence they carry. Types description example: exocytotic vesicles, constitutive vesicle, contains proteins, antibody release by activated plasma B cells, Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Nuclear pore, 3. Rough endoplasmic reticulum. RER, 4. Smooth endoplasmic reticulum, SER. 5. Ribosome attached to RER, 6. Macromolecules, 7. Transport vesicles, 8. Golgi. Apparatus, 9. CIS face of Golgi apparatus, 10. Trans face of Golgi apparatus, 11. System of the Golgi apparatus. Destined for extracellular release. After packaging, the vesicles bud off and immediately move towards the plasma membrane, where they fuse and release the contents into the extracellular space in a process known as constitutive secretion. Secretory vesicles. Regulated vesicles contain proteins destined for extracellular release. After packaging, the vesicles bud off and are stored in the cell until a signal is given for their release. When the appropriate signal is neurotransmitter release from neurons. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Received, they move toward the membrane and fuse to release their contents. This process is known as regulated secretion. Lysosomal vesicles. Vesicles contain proteins and ribosomes destined for the lysosome, a degradative organelle containing many acid hydrolases, or to lysosome-like storage organelles. These proteins include both digestive enzymes and membrane proteins. The vesicle first fuses with the late endosome, and the contents are then transferred to the lysosome via unknown mechanisms. Digestive proteases destined for the lysosome. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Current models of vesicular transport and trafficking. Model 1, anterograde vesicular transport between stable compartments. In this model, the Golgi is viewed as a set of stable compartments that work together. Each compartment has a unique collection of enzymes that work to modify protein cargo. Proteins are delivered from the to the CIS face using copy-coated vesicles. Cargo then progress toward the trans face in copy-coated vesicles. This model proposes that copy vesicles move in two directions. Anterograde vesicles carry secretory proteins, while retrograde vesicles recycle Golgi-specific trafficking proteins. 14. Strengths. The model explains observations of compartments, polarized distribution of enzymes, and waves of moving vesicles. It also attempts to explain how Golgi-specific enzymes are recycled. 14. Weaknesses. Since the amount of copy vesicles varies drastically among types of cells, this model cannot easily explain high trafficking activity within the Golgi for both small and large cargoes. Additionally, there is no convincing evidence that copy vesicles move in both the anterograde and retrograde directions. 14. This model was widely accepted from the early 1980s until the late 1990s. 14. Model 2, Systinal Progression/Maturation. In this model, the fusion of copy I vesicles from the begins the formation of the first CIS. Cystine of the Golgi stack, which progresses later to become mature TGN cystine. Once matured, the TGN cystine dissolve to become secretory vesicles. While this progression occurs, copy vesicles continually recycle Golgi-specific proteins by delivery from older to younger cystine. Different recycling patterns may account for the differing biochemistry throughout the Golgi stack. Thus, the compartments within the Golgi are seen as discrete kinetic stages of the maturing Golgi apparatus. 14. Strengths. The model addresses the existence of Golgi compartments, as well as differing biochemistry within the cystine, transport of large proteins, transient formation and disintegration of the cystine, and retrograde mobility of native Golgi proteins, and it can account for the variability seen in the structures of the Golgi. 14. Weaknesses. This model cannot easily explain the observation of fused Golgi networks, tubular connections among cystine, and differing kinetics of secretory cargo exit. 14. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Model 3, Cystinal Progression/Maturation with Heterotypic Tubular Transport. This model is an extension of the Cystinal Progression/Maturation model. It incorporates the existence of tubular connections among the cystine that form the Golgi ribbon, in which cystine within a stack are linked. This model posits that the tubules are important for bidirectional traffic in the Golgi system. They allow for fast anterograde traffic of small cargo and forward/ or the retrograde traffic of native Golgi proteins. 14, 15. Strengths. This model encompasses the strengths of the cystinal progression/maturation model that also explains rapid trafficking of cargo, and how native Golgi proteins can recycle independently of copy vesicles. 14. Weaknesses. This model cannot explain the transport kinetics of large protein cargo, such as collagen. Additionally, tubular connections are not prevalent in plant cells. The roles that these connections have can be attributed to a cell-specific specialization rather than a universal trait. If the membranes are continuous, that suggests the existence of mechanisms that preserve the unique biochemical gradients observed throughout the Golgi apparatus. 14. Model 4, Rapid Partitioning in a Mixed Golgi. This rapid partitioning model is the most drastic alteration of the traditional vesicular trafficking point of view. Proponents of this model hypothesize that the Golgi works as a single unit, containing domains that function separately in the processing and export of protein cargo. Cargo from the move between these two domains, and randomly exit from any level of the Golgi to their final location. This model is supported by the observation that cargo exits the Golgi in a pattern best described by exponential kinetics. The existence of domains is supported by fluorescence microscopy data. 14. Strengths. Notably, this model explains the exponential kinetics of cargo exit of both large and small proteins, whereas other models cannot. 14. Weaknesses. This model cannot explain the transport kinetics of large protein cargo, such as collaging. This model falls short on explaining the observation of discrete compartments and polarized biochemistry of the Golgi system. It also does not explain formation and disintegration of the Golgi network, nor the role of copy vesicles. 14. Model 5. Stable compartments as cysternal model progenitors. This is the most recent model. In this model, the Golgi is seen as a collection of stable compartments defined by Rab, G protein, GT parses. 14. Strengths. This model is consistent with numerous observations and encompasses some of the strengths of the cysternal progression / maturation model. Additionally, what is known of the Rab-GT parse roles in mammalian endosomes can help predict putative roles within the Golgi. This model is unique in that it can explain the observation of megavesicle transport intermediates. 14. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Weaknesses. This model does not explain morphological variations in the Golgi apparatus, nor define a role for copy vesicles. This model does not apply well for plants, algae, and fungi in which individual Golgi stacks are observed. Transfer of domains between stacks is not likely. Additionally, megavesicles are not established to be intra-Golgi transporters. 14. Though there are multiple models that attempt to explain vesicular traffic throughout the Golgi, no individual model can independently explain all observations of the Golgi apparatus. Currently, the cysternal progression / maturation model is the most accepted among scientists, accommodating many observations across eukaryotes. The other models are still important in framing questions and guiding future experimentation. Among the fundamental unanswered questions are the directionality of copy vesicles and role of Rab-GT parses in modulating protein cargo traffic. 14. Briefeldinner. Briefeldinner, BFA, is a fungal metabolite used experimentally to disrupt the secretion pathway as a method of testing Golgi function. 16. BFA blocks the activation of some ADP ribosylation factors, ARFs. 17. ARFs are small GT parses which regulate vesicular trafficking through the binding of COPs to endosomes and the Golgi. 17. BFA inhibits the function of several guanine nucleotide exchange factors, GEFs, that mediate GTP binding of ARFs. 17. Treatment of cells with BFA thus disrupts the secretion pathway, promoting disassembly of the Golgi apparatus and distributing Golgi proteins to the endosomes and a 16. 17. Gallery. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Yeast Golgi dynamics. Green labels early Golgi, red labels late Golgi. 18. Two Golgi stacks connected as a ribbon in a mouse cell. Taken from the movie. Three-dimensional projection of a mammalian Golgi stack imaged by confocal microscopy and volume surface. Rendered using Imari's software. Taken from the movie. References 1. Pavelkem, Myanor 2008. Golgi apparatus inheritance. The Golgi apparatus, state of the art. 110 years after Camillo Golgi's discovery. Berlin, Springer. p. 580. Do. 10.1000/978-3-211-76310. 0_34. ISBN 978-3-211-76310-0. 2-ABCDE Fabin P.F., Bentivoglio M., October, 1998. 1898–1998, Camillo Golgi and the Golgi, 100 Years of Terminological Clones. Brain Research Bulletin. 47, 31958 10.1016/S0361. 9230, 98, 00079-3. PMID 9865849. S2 SID 2087855591. 3 Golgi C, 1898. Intorno alla struttura del cellule nervoso, PDF. Bollettino della Società Medico Chirurgica di Pavia. 13, 1316. Archived, PDF, from the original on 7 April 2018. 4 AB Davidson M.W., 13 December 2004. The Golgi apparatus. Molecular expressions. Florida State University. Archived from the original on 7 November 2006. Retrieved 20 September 2010. 5 ABCDEFGH Alberts, Bruce, et al. 1994. Molecular Biology of the Cell. Garland Publishing. ISBN 978-0-8053-1619-0. 6 ABCDE Nakanoa, Luinia, August 2010. Passage through the Golgi. Current opinion in cell biology. 22, 44718-2. 10.1016/J.Seb.2010.05.003. PMID 2060540. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 7 Suda Y, Nakanoa, April 2012. The Yeast Golgi apparatus. Traffic. 13, 450510. Do. 10.1000/J.1000/600-0854.2011.01316.x. PMID 22132734. 8 Duran J.M., Kinseth M., Bossard C., Rose D.W., Polishkik R., Wu C.C., Yates J., Zimmerman T., Malhora V., June 2008. The role of GRASP 55 in Golgi fragmentation and entry of cells into mitosis. Molecular Biology of the Cell. 19, 62579-87 do. 10.1091/mbc.e07-10-0998. PMC 2397314. PMID 18385516. 9 Day, KCJ. Kassler, Jason C. Glick, Benjamin S. 2018. Budding yeast has a minimal endomembrane system. Developmental cell. 44, 15672.E4 do. 10.1016/J.Devsil.2017.12.014. PMC 5765772. PMID 293164441. 10 carat ABCD Day KJ, Ster Elin LA, Glick BS, September 2013. A three-stage model of Golgi structure and function. Histochemistry and cell biology. 140, 323949 do. 10.1000/S00418-013-1128-3 PMC 3779436 PMID 2388164 11 carat Campbell, Nilo, 1996. Biology, 4 ed. Menlo Park CA, Benjamin / Cummings. pp. 122, 123. ISBN 978-0-8053-1957-6. 12 carat William G. Flynn, 2008. Biotechnology and Bioengineering. Nova Publishers. pp. 45. ISBN 978-1-60456-067-1. Retrieved 13 November 2010. 13 carat PRYDZK, Dalen KT, January 2000. Synthesis and Sorting of Proteoglycans. Journal of Cell Science. 113 PT2, 2193205 do. 10.1042/JCS.113.2.193. PMID 1063307-1. 14 carat ABCDEFGHIJKLMNOPQ Glick BS, Luinia, November 2011. Models for Golgi traffic, a critical assessment. Cold Spring Harbor Perspectives in Biology. 3, 11/005215. do. 10.1001/Perspect.0052215. PMC 3220355. PMID 2187596. 15 carat Wei JH, Seaman J, November 2010. Unraveling the Golgi ribbon. Traffic. 11/1391 400. do. 10.1011/J.100600-0854.2010.01114.x. PMC 4221251. PMID 2104094. 16 carat AB Marie M, Sana Adar, Afsnsdale H, Sarist J, September 2008. Take the train, on fast. Tracks to the cell surface. Cellular and Molecular Life Sciences. 65, 18 2859-74. do. 10.1017/S008-008-8355-0. PMC 7079782. PMID 1872617-4. 17 carat ABCD D'Souza Scory C, Shavria P, May 2006. ARF proteins, roles in membrane traffic and beyond. Nature Reviews. Molecular Cell Biology. 7, 534758 do. 10.1038/NRM 1910. PMID 1663337. S2 SID 19092867. 18 carat Papanica E, Day KJ, Austin J, Glick BS, 2015. Copy selectively drives maturation of the early Golgi. eLife. Forward to 10.7554/eLife.13232. PMC 4758959. PMID 267098939. External links. Media related to Golgi apparatus at Wikimedia Commons. Categories: organelle's anatomy named for one who described it. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.